So guys, let's proceed with step 2. The step 2 is not very, I'm sorry, the step 2 is not very important step. Why? Because it is a reversible reaction. Just like arrows show, this is a reversible reaction. And reversible reaction in glycolysis are not regulated. Hence, these reactions are not important. Just to tell you what actually happens, I'm going to just briefly explain to you step 2. The enzyme involved is phosphohexose isomerase. Well, as the name suggests, it's an isomerase enzyme, hence a isomerization reaction is occurring. What is actually happening? A glucose is being converted into fructose. We know glucose is an aldose sugar, right? And fructose is a ketose sugar. These both are isomers, but glucose is an aldose and fructose is a ketose. So what this reaction is an aldose to ketose isomerization. This is a reversible reaction, hence it is of no importance. So let's proceed to now step 3. Step 3 is the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Let me write this reaction down. Now this is the reaction. Fructose 6 phosphate is being converted to fructose 1,6 bisphosphate, but the point to ponder is that this reaction is actually irreversible. Irreversible means this is another regulatory point. So the enzyme involved in this reaction is phosphofructokinase 1. What does 1 mean here? Phosphofructokinase 1. Well, phosphofructokinase 1 means that phosphate is being added to fructose 6 phosphate at carbon number 1. That is why this enzyme is called phosphofructokinase 1. This is a phosphorylation reaction, obvious. The bi versus bis. This differences was very important, so I thought I should tell you that bi is converted into bi is that when compound has two phosphates but they are present at the same carbon. But bis means the two phosphates are present in a compound in different carbon numbers. That is why fructose 1,6 bisphosphate. One a phosphate at carbon number 1, one phosphate at carbon number 6. That is why it is a bisphosphate. So, and as we know, it is a enzyme is a kinase. So it actually takes a phosphate from ATP, here ATP, and gives it to fructose 6 phosphate, and converting fructose 6 phosphate into fructose 1,6 bisphosphate. This is a irreversible reaction, hence a regulatory point I told you. But remember, this is the main regulatory point of glycolysis. I told you there are 3 steps in glycolysis which are irreversible and are of regulatory importance. That is, they are acted upon by allosteric regulation and hormonal control. But this step is the father of them all. That is, it is the main important step that is regulated. Hence, what actually happens is that phosphofructose kinase 1 is the main regulatory enzyme, because this enzyme is acted upon by those regulations to stop glycolysis or to induce glycolysis. Now, how does that happen? I will tell you now. This is the allosteric regulation of phosphofructokinase. Well, we know that citrate and ATP, they actually represent the energy status of a cell. So if ATP and citrate is in large quantities in the cell, then that means that cell has already large quantities of energy. So it does not need glycolysis anymore. So the increased concentration of citrate and ATP will inhibit phosphofructokinase 1. But AMP represent energy deficiency of a cell. So if AMP is greater quantity in the cell, then it will activate phosphofructokinase. So glycolysis proceed and produce more energy. All these 3 regulators, they were minor compared to what I'm telling you about to tell you. The fructose 2,6 bisphosphate is the main positive regulator. It should be considered super positive regulator. Why? Because this activates phosphofructokinase to greater extent. If fructose 2,6 bisphosphate is increased in the cell, then phosphofructokinase explodes the glycolysis. That is, glycolysis occurs at extreme rates. So, what is fructose 2,6 bisphosphate? Where did that come from? Well, when glycolysis was occurring, fructose 6 phosphate was being formed. Only minor amount of that was being converted into fructose 2,6-bisphosphate. The enzyme involved was actually a complex of two enzymes. Those two complexes were phosphofructokinase 2 and a fructophosphatase. Well, kinase means transfer of phosphate from one group to another. Well, this for domain or the enzyme actually causes conversion of fructose 6 phosphate to fructose 2,6 bisphosphate. But the second enzyme, that is phosphofructophosphatase, actually converts this fructose 2,6 bisphosphate to fructose 6 phosphate. As we can see now that phosphofructokinase is actually the one which forms fructose 2,6 bisphosphate and activates phosphofructokinase 1, but phosphofructophosphatase is actually what causes phosphofructokinase to be inhibited because it removes fructose 2,6 bisphosphate. I hope I'm clear. Now, what actually happens that phosphofructokinase is induced by insulin? Well, how? Here is it. Here is the explanation. The glucose concentration when in blood increases, it increases insulin secretion. Insulin binds to receptors on cell and cascade of reaction occur, that is intracellular signaling occurs, and protein phosphatases are formed inside of cell. This protein phosphatase causes this complex of two enzymes to be phosphorylated. Hence, domain 1, that is phosphofructokinase 2, is induced. So as a result, I can say that insulin, insulin is causing phosphofructose 2 to be, phosphorus 2,6 bisphosphate to be increased and increased glycolysis. Hence, insulin hormone is actually controlling this point. Well, let me explain to you now the second, how glucagon actually affects. Decreased glucose in blood causes increased glucagon secretion. As a result, cascade of reaction occurs when glucagon bind to receptors in the cell, intracellular signaling occur, cyclic AMP is formed. This second messenger is formed. Hence, domain 2 is induced, that is phosphofructophosphatases. The phosphofructophosphatases actually dephosphorylates the this actually phosphorylates the complex and cause the fructose 2,6 concentration to be decreased. So glucagon is actually inhibiting the glycolysis indirectly. Hence, this point I told you was main regulatory point because it is also being influenced by hormones and also being influenced by feedback regulation. So phosphofructokinase is under allosteric as well as hormonal control, just like I told you. This is why step 3 is the main regulatory step. This is the end of step 3. I hope you understand. Let's proceed to step 4. Well, in step 4, a 6 carbon compound is broken down into 3 carbon compound. How? A fructose 1,6 bisphosphate is converted into dihydroacetone phosphate, which is actually a 3 carbon compound, plus glyceraldehyde 3 phosphate, which is also a 3 carbon compound. Here, let me write down reaction. Here, this arrow actually suggests this reaction is reversible reactions. Hence, it is not of regulatory importance. The results are dihydroacetone phosphatase and glyceraldehyde 3 phosphate. Well, what actually happens that the enzyme involved is aldolase. So let me write that down. Aldolase enzyme. DHAP. What is DHAP? Dihydroacetone phosphate is a 3 carbon structure with phosphate at carbon number 1. This is explanation of this compound. Whereas glyceraldehyde 3 phosphate, which is GA3P, is actually 3 carbon structure with phosphate at carbon number 3. But after this compound is converted into this compound. How? DHAP is converted to GA3P. This is also a reversible reaction. This is part of step 4. The enzyme involved is triose phosphate in the isomerase. Isomerase means isomerization reaction is occurring. Dihydroacetone is converted, phosphate is converted into glyceraldehyde 3 phosphate. Hence, as a result, fructose 1,6 bisphosphate is converted into 2-glyceraldehyde 3-phosphate. This is, I'm talking now net production. You see, first it was only glyceraldehyde 3-phosphate and dihydroacetone phosphate, but dihydroacetone phosphate underwent isomerization and was converted into glyceraldehyde 3-phosphate. As a result, we got 2-glyceraldehyde 3-phosphate. So in this reaction, actually we got two 3-carbon compounds. This is an isomerization reaction. Both reactions are reversible, hence they are not of importance. So this is where I end part 2 of this video. Step 4 is ended.